Biotechnology or recombinant DNA is also called as genetic engineering. If you remember from the previous chapter that we did, in which we understood how DNA passes on its genetic information through transcription and translation, then it makes more sense to understand that in the late 70s and early 80s, scientists developed artificial techniques for making recombinant DNA. A recombinant DNA is pretty much inserting a different genetic nucleotide or changing the sequence on the DNA gene pool, which will result in a different messenger RNA, and which in turn will give you a different protein. Genetic engineering using a bacterial colony is using a procedure in which you amplify the DNA. And amplify the DNA means that genetically you use sufficient DNA for various kinds of experiment and analysis that can be taken on later. In this chapter, we will now understand what biotechnology means. That is the use of actually, I'll go to the next slide to explain to you a little bit more. But on the image over here, you can see these bands and you can see lanes. Firstly, if you notice, this is the first lane, second lane, third, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. There are 13 lanes over here, and there are bands. Each band represents a DNA segment, a molecular weight of DNA based on its electrical charges. So it has actually spliced the long DNA and passed it through this gel and separated them based on its molecular weight and charges. Now, when the bands overlap, then that means that there is something in common over there. So, for example, the E. coli isolates from patients whose infections were not juice related. You can see these two have much similar bands. Then there are the ones from patients who drank contaminated juice. Very similar bands over here. And then the apple juice band. So you pretty much, when you compare the bands, you can compare a specimen of one lane to the other. So what is biotechnology? Biotechnology is mainly the use of microorganisms, cells, or cell components to make a product. Microbes have been used in commercial production of foods, vaccines, antibiotics, and vitamins for years. Certain bacteria were also used during the mining to extract valuable elements from their ore. Now, recently we use the process of genetic engineering, in which we recombine DNA. So here is a picture that I'm going to show you and explain to you as to what's happening up here. Now, firstly, in this cell over here, this is a bacterium cell that has its bacterial chromosome, which is all tangled up and attached to the chromosomal membrane. And next to it is a plasmid. A plasmid is, remember, a circular DNA segment. It's an extra chromosomal DNA segment. And the plasmid has this unique property of making photocopy within the cell continuously. Now, what happens is, in the first step, it says vector such as plasmid is isolated. So the plasmid has been isolated from this particular bacterium cell. Now, on the other hand, there is a pool of gene that is of interest, and within the gene interest, there's a little segment that is the most interest, that's the interest piece that where we want to get out. So now the process is used in which this piece of DNA from the interested gene pool is going to be cut out. Now notice up here you have the DNA that is cleaved and by certain enzyme and then separated. Now the third step is that the gene is inserted into the plasmid. Now the plasmid is circular. So before you can insert the gene in the plasmid actually has to be cut. And that is done by enzymes that are called as restriction enzymes. A restriction enzyme will cut, and then there's another kind of enzyme that are called as a ligase. Let me actually, let me show you. So here is a let me insert a text box and write you these terms over here. Restriction enzymes and ligase. Ligase are the kind of enzymes that helps in connections. So ligases will then join the gene of interest within the plasmid, and it will put it back in the circle. Now, in the fourth step, what you do is you insert the plasmid back into a bacterium. Now, the process of insertion into the bacterium is pretty tedious. You actually have to make holes in the bacterial membrane through which the plasmid can be passed through, and then seal those holes. So it's a pretty complicated process. It's a pretty tedious process that has many steps, and it takes a couple of hours to get through this whole thing. Now, once you have the plasmid inside the bacterial cell, the plasmid is now the vector is now going to clone itself. So the word clone pretty much means making copies of itself. So it will clone, and it will produce many genes that are identical copies. So that's why sometimes when we use the word clone in normal language, we mean that it's an exact copy. This is all taking place within a bacterial cell, is mainly because bacterial cells divide very quickly, and so you can like E. coli divides within 20 minutes. So E. coli is a very cost-effective way to produce many genes of interest, and human growth hormone can be produced genetically in modified E. coli in a very pure and cost-effective method. So there is a very important component of this biotechnology, which is to make selected genes in a population. So now here is a recombinant DNA technology, again showing you how the insertion or modifications of genes can produce desired proteins. So for example, from the previous example that we have over here, coming back on this previous slide, I forgot to tell you that once the genes are of interest are cloned, they are then placed back into a colony to grow, so you have multiple copies of it, and then through another process, those genes are extracted and harvested. So here is, for example, an image in which they have shown you that the genes for example, on the left are the ones for the pest resistance that is inserted into plants, and now these plants that will grow will not be affected by a pest. On the other hand, on the picture over here where you see a ship and an oil spill, here you have gene that alters the bacteria for cleaning up a toxic waste. In the picture over here where you have a man standing, the enzyme amylase, cellulose, and other enzymes are used to prepare fabrics for clothing manufacturers. And in the last picture you have over here is a human growth hormone that treats stunted growth. So pretty much the concept is that a gene from cell of one organism can be inserted into the cell of another organism and expressed in that cell. And these modified genes can then help us get very many variety of useful products. And we use the term genetically modified organisms or products. In the US, it's a lot of food, vegetables and fruits, and are used using these recombinant DNA. In Europe, it's a really big controversy, and they're really there's a big battle about that, and they're not much into recombinant DNA. One of the reasons that the recombinant DNA was introduced was, you may have heard of the golden rice, especially in third world countries where the patent food is rice and there was deficiency of rice. So they used the golden rice, which can be used which can be produced with vitamin D in it, and because the people over there had vitamin D deficiency, and also to produce it in much more surplus amount. So the term, what is a recombinant DNA? A recombinant DNA is a term in which you're inserting a foreign DNA into a bacterial cell. And as I said earlier, you need a restriction enzyme, you need plasmids, ligase, and bacterial host. These are the instrument that you will use. So you should definitely know what is the purpose of each. For example, restriction enzyme is like a scissor. It cuts the plasmid, it cuts the foreign DNA, and it is used to, it's just like a scissor where you cut the vector or the desired gene. Plasmid is again your vector, which is the circular DNA. The ligase are the enzymes that help you connect. And the bacterial host is the host, for example, the E. coli that does it. Here's an example of restriction enzyme for defense against viruses. So a gene that will be introduced to defend against viruses will help in combating viral diseases. Now let's understand what are transposomes. What recombinant DNA does actually takes place within naturally growing cells through transposomes, and transposomes are jumping genes. So they jump from one region to the other, and they give you lots of variety. Researchers, they produce recombinant DNA through plasmids or viruses that attack bacteria, and those are called as the bacteriophages. Example for these are bacteria that make insulin and yeast that help make components of hepatitis B vaccine. So here in this example, you have again shown you the recognition site that is cut by the restriction enzymes. The restriction enzymes, when it cuts a particular site, actually leads a blunt end and a sticky end on the other segment DNA. So the sticky ends are then, so this is considered to be a sticky end, and this is the blunt end. And when you put those two together to make a recombinant, the two sticky ends will stick together to join. And the reason for the sticky end is mainly the nucleotide sequences. Notice how AATTC is cut over here, and then you have the opposite AATTC. So the sticky ends are actually going to have nucleotides that are compatible, and they can attach to each other. What are restriction enzymes? Restriction enzymes are pretty much a special class of DNA cutting enzyme that exist in many bacteria. These restriction enzymes have been there for a very long time in nature, especially in archaea or certain bacteria that live at very hot temperatures. And for example, some of the restriction enzymes are BAM-H1 or EcoR1, and they have been isolated from hot spring bacteria that live in very extreme climates. And the bacteria that are naturally occurring, they used to use them to combat viral infections. Bacterial DNA is unaffected because some is methylated. So methylated is that the bacterial DNA is protected from digestion because the cell methylates, which means it adds a methyl group to some of these cytosine segments in the DNA. And these purified forms of the bacterial enzymes are still used in laboratory today. We use them through cloning, and there's an animation on cloning, and I want you to actually, I'm going to show you how to go in here. You would do, and let's go to the copy hyperlink, and it's going to take us to, I'm going to actually show you where that is. Here is the animation site that you can go to. I can't really have you listen to both because it won't record, but when you come here, you play this, or you can download it for PC, and then let's see, and you can see how these restriction enzymes are specific sequences that are recognized and bind to specific DNA sequences. For example, EcoR1 binds to the sequence GATTC. Later on, you can see how the sticky ends are made by the EcoR1. The sticky ends then can be reattached by another enzyme called as ligase. Notice how it's being attached over here. And then you will have a specific sequence that can be recognized and bind to a specific DNA. So I would like you to go to the site and watch these animations. The other one is also on cloning animation. We can go in there too. And that's going to again show you a site. Actually, this one's not playing right now, so I'm not going to show it to you, but I'll go and fix this. So you should make sure you watch these animations. Now, the last term we want to go over is a vector. I pretty much have given you the definition of vector. A vector is a variety of different types of DNA molecule that are present within the extra chromosomal part of the bacteria, and these are circular molecules, and they actually have regions in which, for example, here is a region that is saying ampicillin resistant or lactose Z making up, and there's an origin site on here. So plasmids or viruses can be attached to the DNA host through a vector. So here are three different these three are the sites of restriction enzyme HIND3, BM, H1, and EcoR1. Vectors are only present in bacteria. They're a circular form of DNA molecule. It's really very important because they protect the DNA of the recipient cell. The DNA of a plasmid is circular, and these plasmids have been used in genetic engineering because they help to be isolated from a bacterial cell. They are useful for genetic markers, and many of them are used today for antibiotic resistance by the enzymes, by the bacterial cells. So that's why we have a lot of bacteria that are resistant to certain antibiotics, because they have the resistant gene inserted into these plasmids. So we're going to stop our first lecture over here, and then we will go into the next lecture, which was going to be about what is the technique that uses these genetic engineering techniques.